In nature, some of the simplest organisms can do something remarkable. They move toward or away from light. This behavior, called phototaxis, is common in algae, bacteria, and single-celled protozoa. It helps them find energy or avoid damage. At the heart of this ability is the eyespot, a primitive, light-sensitive organelle that allows these organisms to detect the direction and intensity of light. Now, scientists are creating synthetic droplets that mimic this behavior. These droplets can respond to light, form eye-like spots, and move across surfaces, all without brains, sensors, or nervous systems. This breakthrough represents a step toward artificial life-like systems that sense and adapt to their environment. What exactly are these synthetic eye spots? They are spontaneously forming patterns inside or on the surface of light-sensitive chemical droplets. When exposed to a directed light source, certain droplets made of responsive oils or amphiphilic molecules begin to organize their internal structure in a way that creates a localized dark spot, an eye spot. This spot forms on the side facing the light, much like the pigmented area in natural microorganisms. But unlike biological cells, these droplets don't have proteins or photoreceptors. The eye spot emerges purely through physical and chemical interactions, such as changes in surface tension, chemical gradients, or light-induced reactions. You might ask, can these droplets really move? Yes, in many cases, once the eye spot forms, it triggers motion in the droplet, often in the direction of or away from the light source. This is synthetic phototaxis. Created without motors or electronics, the droplets move because light changes the internal flow of chemicals or the surface properties at one side of the droplet, generating propulsion through mechanisms like the Marangoni effect. A flow caused by surface tension differences. As a result, the droplet behaves like a simple organism, navigating its environment based on external cues. Is this just a curiosity, or could it be useful? It's both. From a scientific perspective, these droplets give us a powerful model to study the origin of behavior in non-living systems. They show how complex responses can emerge from basic physical rules without the need for genes, neurons, or software. This touches on deep questions in physics, biology, and the study of life itself. From a practical standpoint, these droplets could one day be used in autonomous drug delivery, environmental sensing, or microscale robotics, where simple, smart responses to light or chemicals are needed in places too small or fragile for traditional machines. Have scientists created different versions? Yes. Researchers have developed a range of light-sensitive droplets that use photoresponsive surfactants, polymers, and dyes to form eye spots or move in directed ways. Some change color, some change shape, and some even show collective behavior, groups of droplets aligning their motion like schools of fish or flocks of birds. Others have used droplets loaded with chlorophyll or porphyrins to mimic not just eye spots, but light harvesting behavior. Combining movement with energy collection. You might ask, do these droplets learn or evolve? Not yet in the biological sense, but they can be designed to adapt. For example, droplets can be placed in environments where light patterns change over time, causing them to reorient, split, or stop. With enough complexity added, such as feedback loops, energy storage, or internal reaction networks, it may be possible to create synthetic protocells that exhibit memory-like behavior or selection-based dynamics. These would not be alive, but they would behave in ways that blur the line between chemistry and biology. So what does this tell us? It tells us that lifelike behavior can emerge from simple matter, as long as that matter is arranged in the right way. The formation of synthetic eye spots shows that the basic ingredients for sensing, responding, and moving don't require evolution. They can arise from the interaction of light, chemistry, and physics. It also suggests that phototaxis, one of the earliest behaviors in life, may have emerged not from complex design, but from spontaneous self-organization in droplets. Long before DNA or cells existed, light-sensitive droplets contain photochromic surfactants that switch conformation upon illumination, altering interfacial tension and triggering localized fluid flows. These flows concentrate reactive species into a high-intensity region, forming a visible eye spot of accumulated dye or nanoparticles. The surfactant's isomerization kinetics dictate how quickly and sharply the eye spot appears and dissipates. By tuning the surfactant concentration and light wavelength, researchers precisely control the eye spot's size and contrast. This mechanism mimics biological photoreceptor clustering at the cellular membrane. In a mixture with 0.1 mm azobenzene surfactant, a 405 nm laser spot yielded a distinct eye spot within 2 seconds. It's like using a UV flashlight to activate glow-in-the-dark paint in a small area on a spinning ball. The generated eye spot creates a chemical gradient across the droplet, with higher surfactant activity at the illuminated pole. Marangoni flows drive the droplet toward regions of lower interfacial tension, producing directed movement toward or away from the light source. This phototactic drift replicates microbial behavior without genetic machinery, relying solely on physicochemical forces. Droplet speed and directional persistence depend on light intensity and gradient steepness. Such controlled motility demonstrates that simple chemical systems can exhibit complex navigation. Under a 100 UW light gradient, droplets moved at 10 ohm/s directly up the illumination axis. It's like a soap bubble crawling toward a heat lamp. Because the warmer side has lower surface tension, eye spot formation arises from a feedback loop. Light induces surfactant isomerization, which drives flow that further concentrates surfactant and dye. Mathematical models show this positive feedback leads to a stable, non-uniform concentration pattern only above a critical light threshold. Below that threshold, diffusion dominates, and no distinct eye spot forms. Time-lapse imaging reveals that spots nucleate within 0.5 seconds and stabilize in under 5 seconds. The phenomenon exemplifies how non-equilibrium reactions self-organize into functional structures. When light intensity exceeded 50 UW/mm2, droplets consistently formed eye spots. Below 20 UW/mm2, no spots appeared. It's like water freezing into ice only when the temperature drops below a specific point. Above it, no pattern emerges. Eye spot formation is highly sensitive to the illumination wavelength, as surfactant isomerization peaks at specific absorption bands. By selecting dyes and surfactants with distinct spectral responses, droplets can be programmed to respond only to blue or green light. This spectral tuning allows multiplexed control of different droplet populations in the same field. It also prevents unintended activation by ambient light. Such wavelength selectivity parallels the multiple cone types in animal eyes. Droplets with a coumarin-based surfactant responded robustly to 450 nm light, but remained inert under 550 nm illumination. It's like sunglasses that block UV light, but let visible colors through, unchanged. Turning the light off initiates reverse isomerization of the surfactant, causing the eye spot to dissolve back into a uniform distribution. The erasure rate is governed by thermal relaxation kinetics and can be accelerated by heating or secondary illumination. Complete erasure occurs within tens of seconds, resetting the droplet for subsequent cycles. This reversibility allows repeated experiments without chemical replenishment. It demonstrates a robust cycle of pattern formation and dissolution akin to sensory adaptation. After a 10-second illumination pulse, the eye spot fully disappeared within 30 seconds in the dark. It's like a temporary marker that vanishes when you wipe it away with water. In dense emulsions, upstream droplets cast shadows that influence downstream eye spot formation, leading to coordinated phototactic assemblies. Shadow droplets move differently from directly illuminated ones, creating dynamic clustering patterns. Computational simulations predict wave-like fronts of droplet motion, which experiments confirm under patterned illumination. Such emergent collective behavior mirrors schooling in microorganisms responding to light cues. Understanding these interactions informs design of swarm-like microrobots. Under structured light patterns, droplets formed moving bands as trailing ones followed leaders into illuminated zones. It's like a stadium wave where spectators stand or sit based on the movement of those ahead. The conversion of photon energy into mechanical motion depends on the surfactant's quantum yield and the Marangoni stress generated per isomerization event. Efficiency metrics show up to 5% of incident light power can be transduced into droplet propulsion under optimal conditions. Losses arise from non-radiative decay and viscous dissipation in surrounding fluid. Engineering surfactants with higher isomerization rates and dyes with larger absorption cross-sections improves performance. These studies illuminate the thermodynamic limits of light-driven soft machines. A droplet system with enhanced azo dye achieved propulsion efficiency of 4.7% under 532 nm laser excitation. It's like a solar panel that converts a fraction of sunlight into electricity, with losses due to heat. Eyespot-enabled droplets can serve as light-responsive carriers in microfluidic logic devices where illumination patterns route droplets through networks. By projecting light masks onto chip regions, droplet paths are switched dynamically without valves. This optofluidic routing allows programmable operations such as sorting, mixing, or timed release. Integration with on-chip sensors provides real-time feedback for closed-loop control. Such circuits exemplify how chemical logic can interface with fluidic computation. A microfluidic maze guided droplets to different outlets based on projected light shapes, achieving 95% routing accuracy. It's like railroad switches controlled by semaphore signals that direct trains onto different tracks. Light-sensitive droplets with emergent eye spots offer platforms for studying primitive sensory systems and guiding design of soft robots. Future work aims to embed enzymatic reaction pathways for autonomous energy harvesting and signal processing. Hybrid droplets may incorporate magnetic or pH-responsive elements for multi-stimuli control. Scaling down to sub-micron sizes could enable intracellular sensors that navigate toward light sources. These advancements pave the way for adaptive materials that emulate biological phototaxis in synthetic systems. A prototype soft robot composed of hundreds of light-responsive droplets autonomously aggregated toward a LED array. It's like a school of synthetic fish schooling toward a light beacon in a tank.